Hello everyone, this is Anubhav and welcome to our next lecture in our video lecture series titled Membrane Physiology Muscle. In this particular lecture, we will learn about different types of muscles and look in close detail into the membranes of these muscles and learn how exactly do they function and contract. To start off, we are going to look into the first type of muscle, which is known as skeletal muscle. Skeletal muscles are, as the name suggests, associated with our skeleton and help in our locomotion as well as movement of our body parts. These skeletal muscles are made up of skeletal muscle fibers that run along the length of the whole muscle. These skeletal muscle fibers are made up of cells which are actually conjoined with each other. As we can see right here in this picture, this bulge here is the nucleus of the progenitor cell that made up the muscle fiber. All these muscle cells have already fused to make up the whole muscle fiber. Internally, the environment of this muscle fiber is continuous, and there are filamentous proteins that run along these muscle fibers. These are known as myofibrils. These myofibrils are of two types: myosin and actin. And these are actually the proteins that help in the contraction of our muscles. So to start off, let us understand what exactly is the physiological anatomy of the muscle fiber. To start off, the cell membrane of the muscle fiber is known as sarcolemma. It is the cell membrane of the muscle fiber, and it is the plasma membrane and has a polysaccharide coating on top of it. The sarcolemma fuses with the tendon fibers. So tendons are the part of tissue that helps the muscle join with a bone. So at the elbow joint right here, there will be a connection between the muscle and the bone, and this will be via a tendon, and thus the sarcolemma directly fuses to the tendon. Next, we have the sarcoplasm. Sarcoplasm is the cytoplasm of the muscle fiber, and it contains, as I said, myofibrils. There are also large amounts of potassium, magnesium, and phosphate ions, as well as other enzymes. There are also immense amounts of mitochondria present in the sarcoplasm itself, because the muscle fiber requires huge amounts of energy in order to proceed with the contraction process. And these immense amounts of mitochondria produce huge quantities of ATP, which is the energy currency of the cell and helps in the muscle contraction. Finally, we come to the sarcoplasmic reticulum. Sarcoplasmic reticulum is essentially the endoplasmic reticulum of the muscle fiber. This reticulum surrounds the myofibrils and is very important for muscle contraction, as we will see in the later slides, as it is a storehouse of calcium, which is absolutely essential for muscle contraction. Next, we move on to to know more about what exactly these myofibrils are. So, as I said, the myofibrils are of two types, are made up of two separate filamentous proteins, the actin and myosin. As we can see in this schematic right here, the actin and myosin present in the myofibrils are shown in an enlarged view. The thick filaments, which is right here, is the myosin, and the thin filaments shown by just a line are the actin filaments. These are these are arranged in this way to form what we call as the sarcomere, and this sarcomere, multiple sarcomeres join to form the total muscle fiber. So let us look closely into the myofibril itself. Myofibril has separate parts, as we can see, and these are known as bands. So one thing we have to understand is that skeletal muscle is striated, which means when we see it from outside, we can see separate bands appearing on top of the muscle fiber. And these bands are separated into I, A, and Z bands. To start off, let us look at the I band itself. I band is the conglomeration of only the actin filaments. I bands are the conglomeration of only the actin filaments themselves, and as we can see, as the actin filaments are the thinnest out of the two, I bands are also very thin. Further, the I bands are isotropic to polarized light. So, what does this mean? Polarized light is the light that has its electric field vector vibrating only in one plane. So if a polarized light, if we say a light is polarized at 60 degrees, then the electric field vector of the light coming into our detector has and makes an angle 60 degree with respect to the direction of propagation. But in case this property of light is used to check used to probe various materials and check if they are isotropic or not. What isotropic means is that when we throw polarized light at a particular material, the light that gets reflected back or transmitted through it, we does not have any change in the angular polarization. Which means that if polarized light at 60 degree is thrown upon our particular muscle fiber of a particular eye band, then the polarized light, the light that gets out on the other side will have the same angular polarization. So next we have A bands. A bands are these thicker bands that are present right here, are made out of the ends of the actin filaments as well as the myosin present in the middle, and thus the myosin forms the thicker band that we can see right here. A bands, on the other hand, are anisotropic to polarized light. That is, when polarized light is shined upon A bands, the transmitted light that comes out of the A bands will have a change in the degree of angle of polarization. And as we can see right here, the naming nomenclature of both I and A bands are done based on their nature, on their interaction with polarized light. I bands are named after the isotropics I, and A bands are named after the anisotropics A. Next, we move on to Z discs. Z discs are present at the end of the actin filaments that we can see right here and form this crisscross structure which looks like a Z. This Z structure is present across the myofibril and helps in keeping all the myofilaments in place, so that when contraction occurs, the contraction can occur simultaneously. Now the question appears: there are so many myofibrils and myofilaments present. What exactly is keeping them in position? This is done by a heavy molecular weight protein known as titin. Titin is the black band that we can see right here and helps in putting all the myofibrils in place so that no myofibril can detach from each other. As we have seen in the previous slide itself, there are multiple myofibrils present in our muscle fiber, and if any one of them gets detached or contracts in some other way, that can cause damage to the whole muscle. Titin is the molecule that helps in binding the whole thing together and acts like a molecular rope. Uh, the Z disc, as we can see, at the end of the actin filaments that is shown here, and titin is the what attaches the myofibrils together. Sarcomere is the unit of the myofibril and is taken from one Z disc to the other. So the whole myofibril is actually just the accumulation of various sarcomeres. So one of the sarcomere is present here. The second one is present right here with the Z disc ending here, and similarly it goes on. As we have, if we just this is also shown in this schematic. If this particular Z disc is allowed to, if we had drawn the part on the right hand side, we will see the same mirror image of this sarcomere appearing here. Next, let us move on to the molecular characteristics of contractile filaments. So we have been talking about myosin and actin, but what exactly are these myosin and actin molecules? This is a schematic showing us the myosin. The myosin is a protein that contains two heavy chains and four light chains. The heavy chains form a helical tail. That is, they are intertwined like something like this, and this is known as the tail of the myosin molecule. There are also four separate light chains that are present in the myosin molecule as well, which form the globular myosin head, which is this structure right here. And these are the light chains. The whole myosin filament that we have just seen that forms the myofibril has more than 200 molecules of these myosins. Each myosin has this head portion and also a part of the tail that hangs out of the total hangs out of the total myofibril. This is known as the arm and the head, and together they are known as cross bridges. So they hang out of the myofibril itself like a hook. The cross bridges are axially displaced by 120 degrees. Axial displacement by 120 degree means that if we see the myofibril from this end, we will see that the cross bridges are arranged in a spiral-like manner. That is, they are present 360 degree all around the myofibril, the cross bridges. Further, the myosin head that we are seeing right here has ATPase activity. Next, we move on to the active filaments. The active filament is made up of double-stranded F-actin or filamentous actin molecules. Filamentous actin, on the other hand, is actually a polymer of globular or G-actin. The G-actin can instead bind, has ADP attached to it. And this ADP attached G-actin acts as the active site of the actin filament and interacts with the cross bridges of the myosin that we have just seen. Actin filaments are complexes of actin, tropomyosin, and troponin. What is then a tropomyosin? Tropomyosin is a big molecule, about 70 kilodalton, and has a length of around 40 nanometers. The tropomyosin wraps around F-actin and rests on its active sites. So this, as we can see in this picture, the F-actin is shown by the lighter strand, and tropomyosin binds around it like an intertwined rope and sits atop the active sites of the F-actin. This sitting of tropomyosin prevents actin-myosin interactions, and thus the cross bridges present on the myosin cannot interact with the actin molecule, actin active sites. Next, we have come to troponin. Troponin is a smaller molecule, smaller protein that helps the binding of tropomyosin to actin. It is a trimer of troponin I, which binds to actin, troponin T, which binds to tropomyosin, and troponin C, which acts to calcium ions. When calcium ions are present, as we will see, troponin helps in muscle contraction by the interaction of calcium ions with the troponin C subunit of troponin. Now let us look into how exactly calcium ions interact with troponin. So when active sites of the active filament are normally covered by the troponin-tropomyosin complex. But when calcium ions are released or are present in higher quantity than usual, calcium binds to the TRC subunit of troponin and produces a conformational change. This conformational change uncovers the active sites of actin, which allows actin-myosin interaction. And thus, actin-myosin can interact as shown in this picture right here. But what does actin-myosin interaction entail? How does it help in contraction? This is shown by the walk-along theory of contraction. The myosin head, which was ordinarily just hanging perpendicularly out of the myosin filament, can now attach to the actin active site. So the myosin head will go and attach to the actin active site that is present on the G actin. The head then tilts forward, dragging the actin filament. So the head will move from this position to this position to this position, and the pulls the actin filament while it does. Similarly, as another myosin cross bridge will also pull the actin filament in the same direction. This is known as the power stroke. The head then automatically disengages and returns to its original position. So once it pushes or pulls the actin filament in one direction, it disengages and returns to its original position, just like a boat rower that rows the boat along the river. So once the rowing is done, the row returns back to its original position to repeat the process. Next, the myosin cross bridge binds to a new active site and produces a new power stroke. And with these subsequent power strokes, the actin filament moves or slides along the myosin filament. Next, we come to the source of energy for contraction. Like, what is the exactly helps in the contraction of the muscle fiber? And obviously, the answer is ATP. As contraction begins, the myosin head binds to ATP and cleaves it in using its ATP as activity. And as we know that ATP after cleavage forms ADP and phosphate. Both the products of the reaction, ADP and phosphate, remain attached to the head. The head then extends perpendicularly towards the actin filament but cannot attach it as the troponin tropomyosin complex is already in place. But in the presence of calcium ion, the troponin tropomyosin complex binds. In the presence of calcium ion, troponin tropomyosin complex now binds calcium instead, allowing the actin filament active site to be uncovered. Once the actin filament active sites are uncovered, the head binds to the actin active sites and can then proceed with the whole process. Attachment of the cross bridge to the active site causes a conformational change in the head, which prompts it to tilt towards the arm of the cross bridge, pulling the actin filament along. This is the power stroke, for which the energy was already stored in the form of a spring when ATP was cleaved earlier. So the ATP that was cleaved in the first step, the energy is actually used in this step to generate the power stroke. This tilting allows another conformational change which releases ADP and phosphate, and a new ATP can now bind in its place. This binding now causes detachment of the head from the actin, and the whole process returns to the first step again, where ATP gets cleaved and the whole whole sliding mechanism acts once more. The next cycle begins with the head attaching to a new active site on the actin filament. So once it has pulled the actin filament along, the cross bridge returns to its original position. But as the actin filament has already moved along the myosin filament, a new active site is now open for the cross bridge to bind to and again pull back using a power stroke. Next, we move on to the sliding filament mechanism for muscle contraction. So exactly how does pulling the actin filament along helps in muscle contraction? In the relaxed state, the actin filaments from the Z discs have very little overlap. So this is one Z disc, this is another Z disc, and this is the actin filament from the left one, this is the one from the right one. And as we can see, very little overlap is present there. But as the contraction proceeds, the actin filaments are pulled and their ends start overlapping. into the ends of the myosin filaments, and this is known as the sliding filament mechanism. As we can see right here, as this myosin filament pulls the actin from the right to the left, and this myosin pulls the actin from the left to the right, the actin filaments now start overlapping. And this exactly is what we mean by sliding filament mechanism. This sliding filament mechanism, as you can see, has essentially decreased the length of the whole sarcomere. And this is where the contraction occurs. The sliding mechanism obviously requires energy in the form of ATP, and it occurs in presence of large quantities of calcium. And where does the calcium ion come from? The calcium ion comes from the sarcoplasmic reticulum. So we continue. So with muscle contraction and overlap between the actin and myosin filament. So how does muscle contract? The force of muscle contraction is related to the overlap that we were just discussing in the sliding mechanism slide. The actin myosin overlap is minimum at D in the relaxed state, that is shown right here. Overlap increases till actin filaments overlap all cross bridges, and the sarcomere length is about 2.2 micrometers. That is, once the contraction begins and the filaments overlap, the whole muscles move from the D position to the C position. This is where the maximum tension of the muscle occurs. Further shortening maintains tension till B. So further, if we increase the shortening of the sarcomere, the tension remains the same, but the Z discs have moved even closer. And this is when the actin filaments begin overlapping, and the sarcomere length is about 2 micrometers. Finally, at A, the sarcomere length due to further overlap has decreased to 1.65 micrometers, and the strength of the contraction decreases rapidly. This is when the Z discs have reached the end of the myosin filaments, and the length of the muscle is minimal. And after this, there is a huge drop in muscle contraction strength. This occurs when the actin filaments are finally one actin filament from one Z disc has reached the actin filament has reached the Z disc on the other hand, and now a further overlap along the actin myosin filament of the actin myosin filament cause the filament themselves to crumple along the Z disc, and this decreases the strength of contraction rapidly. And this is shown as a schematic right here. This is the D point where there was very little overlap between actin and myosin. At the C point where there is maximum strength, actin and myosin overlap the most. At the B point, actin filaments have reached their ends and will start overlapping, and the tension is still maintained. But finally, here we can see that the actin filaments have overlapped a lot, and the filaments now have abutted with the Z discs at these ends. This will cause crumpling of the filaments and huge decrease in the strength of contraction. So we'll go through how the general mechanism of muscle contraction. So muscle contraction occurs when a motor nerve innervates muscle fibers, which is innervating a muscle fiber carries an action potential to it. At the neuromuscular junction, or the junction at which the nerve and muscles contact, nerves secrete a neurotransmitter known as acetylcholine. Acetylcholine binds to acetylcholine gated channels on the muscle itself and opens them. The acetylcholine gated channels are cationic channels, and opening of these channels allow entry of large amounts of sodium. And as we have seen in previous videos, that entry of sodium or positive charge into a particular cell allows it to depolarize, and thus sodium ions entering the muscle fiber depolarizes the muscle fiber, initiating an action potential in the muscle. One thing we have to know that this property of polarizing and depolarizing and show an action potential is only shown by two tissues in our body, one of which is the nerve and other is the muscle. As the action potential depolarizes the muscle membrane, this causes flow of electricity into the center of the muscle. So the whole of the membrane of the muscle gets depolarized, and this depolarization wave travels to the center of the muscle. In response to this flow of electricity, the sarcoplasmic reticulum releases large quantities of calcium. Calcium ion, as we have seen, now initiates the sliding filament mechanism for muscle contraction, and thus muscle contraction is initiated. But after a fraction of a second, the calcium ions are now pumped back into the sarcoplasmic reticulum by a calcium membrane pump. And this calcium membrane pump now removes all the excess calcium present, and removal of the calcium causes muscle contraction to cease. And this is the whole process of muscle contraction. Next, we move on to the energetics of muscle contraction. So where is exactly the energy of the ATP supplied for muscle contraction? The energy that is supplied is required for two major processes. First is the pumping of calcium ions back into the sarcoplasmic reticulum from the sarcoplasm. So after the contraction is finished, the excess calcium needs to be sequestered back into the sarcoplasmic reticulum, and for this ATP is required. Next, there needs to be re-maintenance of the concentration gradients of various ions, such as sodium and potassium, across the muscle, which will prepare the muscle for the next contraction by polarizing. The pumping of sodium and potassium concentrations to appropriate values after muscle contraction also requires ATP. But where is the source of ATP? The first source that is present in the muscles is phosphocreatinine. And the high energy bond present in phosphocreatinine resembles ATP, but the energy of the bond is higher than that of ATP, and thus phosphocreatinine can spontaneously release a phosphate group which can reconstitute ATP from ADP and phosphate. So small quantities of phosphocreatinine is present in muscles, which can allow, which once is required, can release the phosphate present in them in order to produce ATP from ADP and inorganic phosphate. But once this storehouse of phosphocreatinine is over, the muscle also has other ways to create ATP, which includes glycolysis of glycogen to produce ATP, as well as the oxidative metabolism via the mitochondria in order to form ATP. Next, we will talk in detail about exactly how the neuromuscular transmission occurs and how the excitation contraction coupling or the contraction of the muscles occur at the molecular level. So first off, let us look at the neuromuscular junction. Skeletal muscle fibers are innervated with large motor nerve fibers, which are shown right here. The nerve endings makes a junction near the midpoint of the muscle, and which is known as the neuromuscular junction. The neuromuscular junction has a schematic shown right here. So the motor nerves comes and makes a junction on the muscle fiber itself. Invaginated membrane is called the synaptic trough. So where the motor nerve makes a contact with the muscle is invaginated like this, and this part is known as the synaptic trough. And the space between the nerve and the muscle is known as the synaptic cleft. The axon, and obviously the axon terminal of the nerve releases synaptic vesicles which contain the neurotransmitter acetylcholine. Next, we'll look into how exactly this released acetylcholine works. So presynaptic nerve terminal, that is the nerve terminal that was coming into contact, which was innervating the muscle fiber, contains acetylcholine vesicles that we can see right here. When action potential reaches the axon terminal, so the presynaptic nerve terminal also has an action potential before the muscle fiber itself. This action potential is due to the brain signaling the particular nerve to move the muscle. So the presynaptic nerve terminal also has an action potential traveling through it, and this action potential, when it reaches the axon terminal, allows voltage gated calcium channels to open, which are shown right here. These voltage gated calcium channels, once they open, allow the entry of calcium ions, and calcium ions, once they enter the nerve, allow the fusion of these acetylcholine vesicles with the nerve membrane, thus emptying the vesicles of the acetylcholine present in them, which is released into the synaptic cleft. The vesicles then empty the acetylcholine present in them into the synaptic space by exocytosis, which is a process of release of vesicles and everything in them into the extracellular. As we can see in this particular diagram, the vesicles, the neural membrane, basal lamina, acetylcholine receptor, and the subneural cleft are all shown. Next, what exactly does the release of acetylcholine do? Acetylcholine gated channels are present near the mouth of the subneural cleft on the muscle side. So these acetylcholine gated channels are present on the muscle. Once two molecules of acetylcholine bind to these acetylcholine gated channels, the channels open and allow the flow of positive ions into the cell. The entry of sodium and the outflow of potassium and calcium along their concentration gradients produce action potential and depolarizes the muscle membrane. So as occurs in the nerve fiber itself, the opening of such a cationic channel allows the flow of sodium along its concentration gradient inside, and the flow of potassium along its concentration gradient outside of this. This causes a net decrease or net depolarization of the membrane itself, and this initiates an action potential. After signal transmission, once the work of acetylcholine is done, it is degraded by the enzyme acetylcholine esterase. Small amounts of acetylcholine esterase diffuses out of the subneural cleft as well as small amounts of acetylcholine that did not bind to such gates also diffuses out of the subneural cleft and is then degraded elsewhere by acetylcholine esterase, and thus acetylcholine is removed from the subneural cleft after its action is done. Now we come to the concept of end plate potential. End plate potential is the potential of the muscle fiber itself after an action potential has been initiated or the nerve has tried to stimulate it. The sudden entry of sodium ions cause potential inside the fiber to increase. The increase of the muscle, the increase of the resting potential of the muscle from its normal value to a 50 to 75 millivolt increase initiates the action potential and is known as the end plate potential of the muscle fiber. As we can see, we have a diagrammatic representation of the end plate potentials that can be generated in muscles. So to start off, we have the end plate potential B, which is strong enough to open enough sodium channels to initiate an action. So as we have seen in previous videos themselves, that there is a particular threshold that needs to be crossed for the action potential to be initiated for the whole membrane. And this end plate potential can produce such a depolarization or allows as many sodium ions to flow into the muscle fiber itself that can allow the continuation of the action potential. But in case of end-plate potentials A and C, we can see there is stimulation, but it is not enough to generate a full-blown action potential. Such a case is because the muscle can be impaired, and in case of end plate potential A, the muscle fiber itself is poisoned with the curare poison, which does not allow it to initiate a full-blown action potential. Curare acts by blocking acetylcholine channels by competing with acetylcholine. So the acetylcholine gated channels that we have seen, they are binded by curare and prevents binding of acetylcholine with it. Such action will then cause no muscle contraction even if nerve stimulation is present. And this is what we can see in end plate potential A. In end plate potential B, muscle fibers are affected with the botulinum toxin, which also prevents action potential to occur, but its mechanism of action is a bit different. It prevents acetylcholine release from the presynaptic nerve fiber itself, and this in turn causes a no action potential to occur on the muscle cell. In addition to these particular drugs, such as the particular poisons that can affect the muscle, there are other drugs that can also do the same. These include stimulating drugs such as nicotine, which bind to the muscle, bind to the acetylcholine gated channels in place of acetylcholine, but instead cannot be degraded by acetylcholine esterase. This causes the muscle to go into a constant contraction or a spasm. So let us move on and look at excitation contraction coupling, or how exactly the muscles contract in response to excitation by nerve fibers. Depolarization of the muscle fiber membrane travels deep into the muscle via transverse tubules. Transverse tubules are extensions of the extracellular membrane, extracellular environment into the muscle fiber itself. So if we can see the muscle fiber as the diagram shows, if a membrane depolarization occurs on the surface itself, it will never travel into the center or the belly of the muscle. Thus the muscle fibers present in the middle of the muscles cannot be excited via a membrane potential. This problem is solved using transverse tubules, which are essentially the extension of the membrane deep into the muscle. And as the depolarization occurs on the muscle itself, the transverse tubules also take the depolarization deep into the muscle belly and excite the muscle fibers present there. T-tubules are internal extension of the cell membrane. The sarcoplasmic reticulum is connected to T-tubules via the terminal cisternae. So these are a network which connect the transverse tubules to the sarcoplasmic reticulum as shown here. The SR or sarcoplasmic reticulum also extends longitudinal tubules which surround contracting myofibrils. The sarcoplasmic reticulum is closely associated with the contracting myofibrils and surrounded from all sides. Next, we will look into the role of sarcoplasmic reticulum and calcium ions. As action potential causes current flow into the sarcoplasmic reticulum from the T-tubules, it opens a large number of voltage-gated calcium channels. So the sarcoplasmic reticulum is essentially a storehouse of calcium ions, and flow of and depolarization of its membrane opens various voltage-gated calcium channels along it. The calcium ions now flow into the cytoplasm and enable myofibril contraction via the mechanism, via the troponin tropomyosin mechanism that we have already looked into. Once the contraction is over, calcium ions need to be sequestered back into the sarcoplasmic reticulum fast. And this is done using active calcium pumps which sequester calcium ions back into the sarcoplasmic reticulum. Sarcoplasmic reticulum also contains a protein known as calsequestrin, which can bind to calcium in large amounts and helps to remove excess calcium present in the sarcoplasm after the contraction has occurred. When an excitatory pulse occurs, the calcium concentration can increase from less than 10 to the power minus 7 moles, 7 molar, to 2 into 10 to the power minus 4 molar. So close to a 2000-fold increase in calcium contraction. But this calcium pulse lasts only about 1/20th of a second. And if we thus if we need continued contraction of the skeletal muscle, then this requires a series of pulses which will be placed so that the calcium concentration can be maintained in the sarcoplasm and the skeletal muscle continues to contract. Next, we move into fast versus slow muscle fibers. So there are two types of skeletal muscle fibers: the fast fibers and slow fibers. There are a lot of differences between them that we will look into right now. Fast fibers are large fibers which are required for great strength of muscles. These include muscle fibers present on our body, such as our triceps and biceps, which move large parts of our arm and thus require great strength of contraction. These fast fibers also contain an extensive sarcoplasmic reticulum for fast release of calcium, which helps in their contraction. Next, they also contain large amounts of glycolytic enzymes, which help in the release of energy via glycolysis. And also, this glycolysis is the main source of energy for fast fibers via ATP. But instead, they have less extensive blood supply and fewer mitochondria because oxidative metabolism itself is secondary in fast fibers. Slow fibers, on the other hand, are smaller fibers which are innervated by smaller nerves. There is extensive blood supply and increased number of mitochondria present in slow fibers which support the oxidative metabolism. So slow fibers essentially work via oxidative metabolism, whereas fast fibers produce energy via the glycolysis mechanism. Slow fibers also have large amounts of myoglobin, which is a substance similar to hemoglobin, and myoglobin helps in conduction of oxygen to the muscle fiber themselves. Next, after all this discussion, we are finally over with skeletal muscle fibers, and we look into smooth muscle fibers. Smooth muscle fibers are smaller muscle fibers with a diameter of 1 to 5 micrometer and a length of about 20 to 500 micrometer. Smooth muscle fibers are not related to our skeleton or anything, but are related with respect to glands and visceral organs present in us. So they essentially line various glands and visceral organs such as the urinary bladder and act and form the wall around it. The smooth muscle fibers are divided into two types: multi-unit smooth muscle fibers and unitary smooth muscle fibers. Smooth muscles. The multi-unit smooth muscles are discrete, separate smooth muscle fibers and are independently innervated. So these are closely present. The multi-unit smooth muscle is made out of closely present smooth muscle fibers which are individually innervated. So any signal coming from a nerve can individually excite particular smooth muscles, or as occurs, can excite a smaller part of the smooth muscle fiber itself and not excite the other part of the smooth muscle. And the other type of the smooth muscle is the unitary smooth muscle, which has a mass of 100 to 1,000 fibers that contract together. Like, for example, the urinary bladder that can contract at once. The depolarization is transmitted via gap junctions. So, what are gap junctions? Gap junctions are particular junctions present between muscle fibers that allow the flow of sodium ions through them. So the muscle fibers themselves are connected by these gap junctions, and the internal environment is continuous. And thus, the depolarization wave, which excites one muscle fiber, can also excite the others as well. And this is how unitary smooth muscles function, where the excitation wave can travel through the whole muscle itself via these gap junctions and depolarize and contract the whole muscle. Next, we move into the mechanism of contraction in smooth muscles. Smooth muscles do not have the striated appearance of the skeletal muscles. This contains instead dense bodies to which actin filaments are attached. So, what does this mean is that smooth muscles have a very the name smooth muscle itself tells us that smooth muscles have a plainish looking appearance with no stripes, such as the skeletal muscle. Instead, they have these dense bodies to which the actin filaments are attached. So the arrangement is similar to the sarcomere, but instead of the Z discs, we have dense bodies present here. The myosin filaments are also different in the smooth muscles and are have side polar cross beaches. Side polar cross beaches mean that the cross bridges are present on either side, like 180-degree opposite to each other, and do not cover the whole myosin filament in a 360 spiral manner as in the skeletal muscles. The myosin can simultaneously pull two actin filaments in opposite direction. Since we can imagine that the myosin is present and actin filaments are present on either side of it. As we'll see in the following slide, there is schematic of it. When the myosin contracts, it pulls oppositely present actin filaments towards each other. The smooth muscles further can contract about 80% of their length instead of the only 30% of skeletal muscle. The reason of this is the absence of any Z discs and any striated bands that will prevent further contraction. So this is how what the side polar myosin filaments looks like. So the myosin filaments are present on either side 180-degree across each other. So there are no cross bridges present above the plane or below the plane of the myosin filament. And this is an example of a skeletal muscle fiber skeletal muscle fiber myofilament whose cross bridges are present on all directions. So when a skeletal muscle fiber tries to contract, it will pull the actin filament on its left side towards the right and the actin filament on the right towards the left, but it is restricted by the presence of the Z disc, which will finally crumple the myosin and prevents further contraction. But due to the presence of dense bodies as well as the side polar myosin arrangement of the smooth muscles, if the smooth muscles start contracting, these actin filaments can move to a huge length along the myosin filament before they are restricted to move further. And thus we have this large contraction present in smooth muscles as opposed to skeletal muscles. Further, we have further differences between the smooth muscles contraction and skeletal muscle contraction itself. The smooth muscles have a slow cycling of myosin cross bridges. So the sliding mechanism does not occur very fast in smooth muscles. The power stroke cycle is much slower in smooth muscles. It is about 1/10 to 1/300 of the skeletal muscles. This slow cycling can be seen as a disadvantage at first, but is actually this increases the fraction of the time the myosin heads remain in contact with the actin active sites. And the contact time between the actin active sites and the myosin cross bridge head is what determines the force of contraction of the muscle. And this slow cycling does indeed help in greatly increasing the force of contraction of smooth muscles. Further, the energy required to maintain this slow cycling is very less, about 1/10 to 1/300 of the energy required by skeletal muscles to maintain the same tension. So, because the one cycle consumes 1 ATP, slow cycling overall thus consumes less energy in the smooth muscles. Further, there is a slowness of contraction and relaxation of the smooth muscles. The whole muscle requires 50 to 100 milliseconds to relax after excitation, which is 30 times as long as skeletal muscles. So the movement of so the return of smooth muscles to their original resting state requires a way longer time than skeletal muscles. And finally, the force of contraction. The maximum force of contraction of smooth muscles is way larger than that of skeletal muscles. This is due to the first one that we have discussed, as the myosin heads remain in contact for longer periods, which is the only determining factor regarding the force of contraction. The max force of contraction of smooth muscles is very much greater than that of skeletal muscles. Now we look into the role of calcium ions in smooth muscle contraction and what exactly is the mechanism of smooth muscle contraction. Smooth muscles do not have the molecule troponin. Instead, they contain the protein calmodulin. Calmodulin also can bind to calcium. The calmodulin calcium complex instead acts differently than that of our skeletal muscles. Calmodulin calcium complex activates another protein known as myosin kinase, which is a phosphorylating enzyme. One of the light chains of the myosin head is known as the regulatory chain, and this is phosphorylated by the myosin kinase. This phosphorylation instead allows myosin actin attachment, giving rise to the muscle contraction. So the work that was getting done by troponin binding to calcium instead is done by the calcium calmodulin complex through the myosin kinase protein. As calcium concentration drops, the another protein, myosin phosphatase, splits the phosphate group from this regulatory chain, which allowed the myosin actin interaction, and this stops the cycling of the myosin cross bridges, and allows the contraction of the muscle. Next, we move on to the latch mechanism of smooth muscle contraction. So once full contraction is achieved in smooth muscles, energy required to maintain the full force of contraction is way less than the initial level. So once the full contraction has been achieved, maintenance of the same requires very less energy. As low as 1/300th of the energy required by skeletal muscle fibers for sustaining the same force of contraction. This low energy consumption to maintain high levels of contraction is known as the latch mechanism of smooth muscles. This latch mechanism has been tried. It has been tried to explain the latch mechanisms via various ways, and one of the possible explanations are given here. As the activation of myosin kinase decreases, as the calcium ion concentration decreases, the cycling frequency of the myosin cross bridges also decrease. This allows myosin heads to remain attached to the actin filaments for longer. As the cycle itself is getting slower and slower till it stops, the myosin heads have enough time to remain attached like hooks to the actin filaments. The number of myosin head attachment actually determines the force of contraction. And this, as the myosin heads are remaining attached to the actin filament itself, this tension is maintained or latched. So, like the latch on our doors, once the latch is put down till it is removed again, the force which blocks the door remains. And similarly, once the myosin head has been latched onto the actin filaments, till it detaches, the whole full force of contraction is maintained. Minimum energy is further required in order to maintain this full force of contraction. Only in the rare case a head detaches. So if the head detaches and comes back to the original position, another ATP is only required to bring it back to the attached position. And this is the only energy consumption that is going on. Otherwise, all the other heads are remaining attached and maintaining the full force of contraction. Next, we move on to another characteristics of smooth muscle known as stress relaxation. Stress relaxation of smooth muscles is is a characteristic shown by smooth muscles, especially in visceral hollow organs, which are stretched or shortened, but have the unique property to return to their initial force of contraction within seconds or minutes. For example, when the urinary bladder suddenly expands, the stretching causes increase in pressure in the lumen of the bladder. In the next 15 seconds or so, the pressure again returns back to the original level and more urine can flow into the bladder. On the other hand, when pressure drops in the lumen, the pressure falls low at first, but then again rises back to the original level. This is known as stress relaxation and reverse stress relaxation, which means that if the smooth muscle is stretched or relaxed, after a while the lumen of the smooth muscle will retain its original pressure without any without any further change in volume. This allows various hollow organs like the bladder, our stomach, to stabilize their contents. This phenomenon allows the hollow organs to maintain the same amount of pressure inside the lumen despite long-term changes in volume. Finally, we come to control of smooth muscle contraction. Neuromuscular junction of autonomic nerve fibers diffuse on top of smooth muscle fiber sheet. Highly structured neuromuscular junction like that we have seen in the case of skeletal muscle fibers do not occur. Instead, a diffusion of nerves across the smooth muscles occur. Autonomic nerves release acetylcholine and norepinephrine as neurotransmitters from different nerve fibers. When acetylcholine excites a nerve fiber, ordinarily norepinephrine will inhibit the nerve fiber and vice versa. Skeletal muscle fibers, on the other hand, only secrete acetylcholine, as we have already discussed, whereas smooth muscle fibers can release the nerves exciting the smooth muscle fibers can release acetylcholine, norepinephrine, and other substances, neurotransmitter substances as well. So that brings us to the end of this particular slide. So in this particular slide we have discussed the membrane physiology of muscles, discussed the two types of muscles, the skeletal muscles as well as smooth muscles, and looked into how exactly they function. In the next slide, or the next lecture, we will look into the heart and the cardiac muscle. Thank you for listening to this video.